I'm very happy to have Dominik here with us as one of great friends and colleagues presenting paper Moscot on application of OT, scalable OT, let's call it, to both spatiotemporal analysis of single-cell data. We are very excited to have him here to present the work, finally published in Nature after a long while of back and forth. So congratulations. And without further ado, please take it with your slides.
Thanks a lot for having me, and thanks everyone for joining. As I said before, just please feel free to ask any questions in between. I think, Shine, you will likely moderate that. So whenever you would like to, just shoot. I'm going to present mainly the Moscot paper, which was published 2 months ago in Nature. It's about scalable optimal transport, as Shaheen mentioned already. I'm going to walk us a little bit through optimal transport in general, because I don't think everyone is familiar with it. And then afterwards highlight the biological findings which we had in the course of that paper. So why is optimal transport actually relevant in single-cell genomics? This is due to mainly that distributions of cells have to be realigned, because sequencing technologies are destructive. So for example, whenever we look at perturbations, we have the control distribution and the perturbed distribution, but it cannot be the same cell. So we cannot measure before, for example, some perturbation and after that perturbation. And hence we have to somehow align them. Similarly, due to biotechnological constraints, we can only measure a limited number of modalities at the same time. Yet, to get a more comprehensive view of the tissue of interest, for example, we would like to have multiple views of our samples, and hence we have to realign those samples. And now there are obviously multiple ways to realign samples or to match distributions, but there's one geometric way which is probably the most natural one, which is optimal transport. And optimal transport is always concerned with aligning, mapping, matching, all sort of synonyms, distributions. And therefore, let's consider 2 sets of data points, can be cells. One is the source distribution in red, one is the target distribution. And we could now guess and try different matchings. So these are just 2 random ones, but we will get very many. We don't want to walk through all of them now. In fact, only given those 6 data points on the source and the target distribution, we already get 720 possible ways of matching those data points. If we talk about single-cell genomics, we normally have nowadays, luckily, many more than only 6 cells, and hence we need to find a way how to naturally match that and how to quantify that. So the question is, how do we choose the best coupling? And therefore, we quantify the cost of each coupling. Therefore, we look at a distance or a cost. That's also a synonym. This can be any distance. It could be a Euclidean distance. It can be graph-based distances, whatever you like. And look at how far each data point in the source distribution is from each data point in the target distribution. And then we want to minimize the overall cost, hence we want to match points which are close together. So we look at all these pairs of connected points, look at their distances, and want to make sure that each data point in the source distribution is mapped to one data point in the target distribution. And here, if we take as a reference the Euclidean distance, x1 and y6 are a natural pair because they're close by, and we can fill that element in the matrix, because for each row, which corresponds to a source data point, we want to find a corresponding data point in the target distribution, and hence we set that entry ij, in that case, the matrix of row 1 and column 6 to 1, or basically to some entry, which indicates that we want to match those data points. Now, we can quantify that cost, which is just the sum of these distances. But this problem is actually very difficult. And indeed, if you think about it, we now had another Y7 on top, we couldn't even perform that matching. Hence, we need to go back to a relaxation. It's called Kantarovich relaxation. Or indeed, we actually even want to promote fuzziness in the optimal transport matrix, which means we want to encourage stochasticity of the optimal transport matrix. This is quantified by the entropy. So here you can see that now we measure the total cost as still the sum of all the green arrows, basically. So how expensive it is to transport all of these samples. But at the same time, we want to encourage high entropy. And this epsilon parameter determines how fuzzy our transport matrix will be. So if epsilon is infinity, we actually don't care which pairs are matched in the end, because in the end, what we will get is we want to get the matrix, the transport matrix, which is here this stochastic mapping, which is as fuzzy as possible. And this is just the outer coupling. So basically the same entry everywhere in the matrix. So this epsilon just tells us how stochastic is our matrix, and it's a hyperparameter to be set. But luckily, in that case, the single hyperparameter which we considered. And yeah, this also leads to computational advantages. In fact, if we don't consider the entropy regularized version, then the problem is very, very hard to solve. While if we now introduce this entropic regularization, which again, as I said, promotes the fuzziness or stochasticity of the transport map, and this actually renders this problem a convex problem, which is very nice because we know we will converge to this solution. Now, there has been prior work quite a bit in single-cell genomics about optimal transport, but we realize there are a few limitations. For example, in terms of modalities, researchers mainly considered unimodal data, but with progress also in biotechnology, we often have now paired readouts, or in general, multiple modalities. And hence, we thought there's potential for improvement to extend also to multiple modalities. Then another very relevant point is scalability of optimal transport algorithms, because optimal transport is very, very expensive in terms of computation and in its original formulation. In fact, it's quadratic or even cubic in memory, and at least quadratic in time. And if you think about large atlases where we have hundreds of thousands of cells, this is just prohibitively expensive, such that we cannot even run it because we don't have the compute to execute that. I'm going to show a little bit later what that actually means. And finally, we want to make our tools accessible to everyone, for biologists and computational biologists to be able to use these actual powerful optimal transport algorithms without having to dive into the details. And hence, we wanted to provide easy access to everyone. Now, as I said, we try to resolve these limitations by looking at multimodal data, and we actually also showed that it improves these mappings just because we get a more comprehensive view of the cellular state. At the same time, we make these optimal transport algorithms across our problem scalable, reduce it to linear time and memory complexity. I'm going to talk about that in a second, how we do that. And finally, we provide a user interface which single-cell biologists are familiar with, which is closely tied to the SCVerse ecosystem, this community of which collects tools and tries to make everything as accessible as possible. So how do we make everything scalable? We briefly discussed that before going into the biological examples. So first of all, we build upon optimal transport tools, OTT-JAX, which is a Python toolbox for optimal transport applications. It implements very many tools. Please feel free to check it out. But one of the advantages is definitely that it implements on-the-fly cost matrix computation. What does that mean? As I said before, we have to consider distances between point clouds. And actually, if you instantiate that cost matrix, you see that we already have like a complexity of n squared, or n times m, where n is the number of data points in the source distribution and n is the number of data points in the target distribution. But if we look at hundreds of thousands of cells, this is prohibitively expensive already, and hence you can actually just do that on the fly. This already reduces memory complexity to linear time while maintaining the exactness of the solution. Additionally, we support GPU acceleration because optimal transport always breaks down to basically some matrix vector multiplications. It's very suitable for being executed on GPUs. And very importantly, we leverage recent low-rank approximation schemes of optimal transport solvers. In particular, that's Maïs Guédon, another student of Marco Couturier, who really pushed these developments, and that allows us to get to linear time and memory complexity across all applications. So what does that mean in practice? We compared moscot and the low-rank version of moscot to Waddington OT. So we can see that with Waddington OT, you cannot go beyond 75,000 cells, even on a server, because it's just, requires too much memory due to the quadratic computational complexity. At the same time, with moscot, we can easily go to 300,000 cells without any problems and could even go further, even on a laptop. And in terms of runtime, that means for 75,000 cells, it almost takes 2 hours to run a Waddington OT, whereas with moscot, we have 2 minutes. And even if we go to 300,000 cells, it's solvable within 15 minutes. So that's just a few examples of how relevant that is. Yeah.
Do you mind if I ask a question here?
Sure.
This is beautiful. I love these types of algorithmic improvements that just make things usable and nice. But I'm also wondering why there's this gap, in terms of runtime, where the low rank approximation is actually more, costly than the full rank.
Yeah. So the low rank approximation is a different algorithm than the full rank algorithm, or like the full rank optimal transport solution. So in fact, it basically reduces the optimal transport matrix into a factorization of 3 matrices. And hence that requires, if you don't have many data points, it requires more multiplications because you just have more matrices to optimize over. You have 3 instead of 1. And at the same time, it's also a little bit about convergence. So because it's not convex anymore, it's just a different optimization scheme. Still some sort of precondition gradient descent, but it's just a little bit different. But it really pays off once you get beyond 75K cells.
Is there any intuition for like the choice of 3 matrices? Because I mean, to do low rank approximation, you would only need 2 typically, and if it's an optimization objective on the full matrix, that should be, I guess, sufficient. Are there limitations of the method that mean that we have to use 3, or there's some? I guess I'm wondering, right? Because
yeah. So there are actually different low rank approximations in general. So just last year at NeurIPS, there was another one. So this is just another one proposed by Maïs Guédon, which we build upon. So yeah, intuition, I don't think so. So basically you project it down, you maybe solve it in a lower dimensional space, and then you project it up again. That helps, but I don't know how much intuition that conveys.
Okay, sure. Yeah, maybe I'll Okay, I'll see if it's answered later. Yeah, I'm not sure I have the full intuition yet, but
maybe if you go, I don't know if you have slides on the like methodological details of the low rank, that might help. If not, we can keep it for later.
Yeah, I don't have it in that presentation. Maybe we can discuss that later.
Yeah, sounds good. Thank you.
Cool. Right, so in terms of applications, we now consider 4 different use cases in the course of this presentation, which is once trajectory inference for multi-omic data. Once we map dissociated cells to their spatial organization, we then align spatial transcriptomic slides, and finally, we have a novel spatial temporal trajectory inference method. So with moscot, we looked at the development of the pancreas. Why this is interesting, I think there are very, very many reasons, but I think most of us know that diabetes is a disease with its origin in the pancreas. So it's basically about beta cells, which we can also see here on the right-hand side. And we want to understand better how these cells evolve. And therefore, we captured the marine pancreata at 3 different time points. With multiome, hence we have paired gene expression and attack measurements. We can already see on the UMAP that they're fairly overlapping, those distributions, or like these cellular measurements across these 3 time points, but still there's some dynamic happening. So we do see a distributional shift. We then created a joint embedding of gene expression and attack using multi-VI, so like some VAE-based representation incorporating both modalities, and created a cost matrix based on heat diffusion on a graph. So this is actually very relevant. I'm not going to go into detail here, but we have it in the paper. Here, just the Euclidean distance wouldn't work. So it's very relevant which cost function we choose, because obviously optimal transport doesn't know about the true dynamics of your data. So sometimes it's a little bit difficult. That's definitely a limitation of optimal transport to choose the correct cost function. So once we've done that, we can now look at which cell type evolves into which cell type. So what we do is basically just aggregate the transport matrix to the cell type level. And we can then see that mature acinar cells are basically only mapped to mature acinar cells for a good reason, because they are differentiated cells. So we don't expect them to develop or differentiate any further. In the second row, you could see immature acinar cells are mainly remain in their cellular state. So they basically, in time point 15.5, will still be immature acinar cells, but some of them will evolve into mature acinar cells, which is also something which we would expect, because immature acinar cells, as the cell type name suggests, will develop into mature acinar cells. So we can see in general, yeah, sorry.
So quick question, you talked about Euclidean distance not always working as the grand cost, but also that brings up another question, because it seems that you use that in a pure VAE embedding space, right? Where it does not have any component of the loss to encourage a metric space when you're training your model. Would that change if we use models like the similarity that recently came out with the the triplet loss, and like the space is like? So how much that embedding space have an effect? Because again, we usually do in the original space, we want to do some space, either PCA, VAE, or some.
Yeah.
How much of a role that has before running your OT on it?
Yeah, that's definitely a good point. So if you can encourage your latent space based on which you want to compute the distances if you want to encourage it to basically be or follow sort of the manifold of the cells, then that's definitely a good way. Actually, there is a paper also from our lab where you encourage the latent space to be globally sort of linear, or follow the Euclidean metric, and there it definitely helps to compute the cost matrices or the distances based on that metric. So it definitely matters a lot. Here we decided to use the multi-VI embedding, but take the other option, which is just adapting the cost function instead.
There is another question also from Ziv, which is, are all matchings one-to-one? If not, any comment on the number of matching for both time points?
Are all matchings one-to-one on the cellular level? Probably that's the question.
Yeah. Yeah, so the exact question is, are all matching, and you can see it in the Q&A in the bottom as well, are all matching one-to-one? If not, any constraints on the number of matchings for both time points? I think you may touch on some of the unbalanced version, which talks about some of the constraints later, which I think is related to this one. But I wanted to bring up the...
Yeah, so if we talk about like one-to-one matching, to what extent cells are matched one-to-one, as we saw before, because we use we solve the entropy regularized optimal transport problem due to these computational advancements, there's no one-to-one matching, and we can steer the stochasticity or the fuzziness of the optimal transport matrix with a hyperparameter, which is epsilon. And now just maybe empirically. So first of all, you cannot really say how many cells each single cell is mapped to, because the probability of mapping or of the matches of one cell or being of one cell in the source distribution being mapped to one cell in the target distribution is not constant across different cells in the target distribution. So the conditional probabilities are not uniform, and hence you cannot really count how many matches you have, because there's not it's not a binary thing, it's a probabilistic matching.
Yeah.
But if you say what are, let's say, entries which are not negligibly small, then we might talk about roughly 10 to 20 cells. But it's a very very rough estimate. And also obviously it depends a lot on the stochasticity and everything.
Yeah, so I think a follow-up to that is, how do you get figures like the ones on the right here, where you do seem to show some sort of matching?
Sorry, can you repeat that, please?
Right, so the figures on the right here, right? You have showing the change between cells, between different time points, and typically you'd get a figure like this by assigning a cell type label to both the before and after time points, and then computing the flow between these two things. But you're saying that you don't actually have this one-to-one mapping. So I'm wondering if in this maybe specifically the flow diagram on the bottom here, right?
Yeah,
is computed from the distributions over cells in the for each cell under the OT, or is this actually assigning a label and then computing the flow, right?
Okay. Yeah, no, this is just an aggregation of the transport matrix. So basically you would integrate over all cells which correspond to that specific cell type. So on maybe this is a little bit misleading here, that the matrix which we see on top seems like, for example, the first row only has one match. But this is an aggregated view. So basically you look at all mature acinar cells, where are they mapped to, and each single mature acinar cell in the early time point will be mapped to multiple mature acinar cells in the later time point. But because we integrate over them, more like differentially speaking, we aggregate them, we don't mind, or we don't see that each single cell of a mature acinar cell in the source distribution will be mapped to multiple cells in the target distribution, because it's an aggregated view. That's important. It's not the output of the it's not the output of the optimal transport solution. This is an aggregated view, because the output of the optimal transport problem is a bistochastic matrix, which has the number of cells in 14.5 dimensions times the number of cells in 15.5. Yes. So it's roughly 20,000 times 20,000, and it's very hard to visualize that or conclude anything from that.
Yeah, and also because these rows and columns always have to sum to 1 over, again, without any prior 1 over number of cells, that's basically a norm 1 constraint. So that usually looks like more sparsity inducing as well. But also I think it would be nice if you show with different values of epsilon. So as you change the epsilon, which induces entropy, you will see more non-zeros. Whereas with, without enough entropy regularization, you see very much there more matching matrices coming out.
So to understand the figures on the right, it's this is actually a property of the method, not a prediction from the OT problem, right? you're saying you're visualizing the transition matrix.
Correct. Which is the output. The transition matrix is the output of the optimal transport problem. We always visualize this mapping, and the mapping is a bistochastic matrix. And what we see here are basically visualizations or aggregated views of the output of the optimal transport problem. Right. And basically in the bottom, you can see a subset of the cells, which are the endocrine cells. So those endocrine cells and their progenitors. So those cells which produce hormones and hence are particularly interesting in the context of diabetes, for example. So what we can do here is we can, for example, look at epsilon cells. And epsilon cells, as I said, they produce a hormone which is called ghrelin. And we look at their distribution at time point 16.5. And then we can compute their ancestors. So basically, what does moscot predict? Which cells does moscot predict to be ancestors of epsilon cells in the latest time point? And then we can actually even look at the ancestors of the ancestors. And that way, we can hypothesize or also look at these transitions on a single cell level. So here, the coloring is according to the probability of each single cell being an ancestor of any epsilon cell. And this way, again, you can roughly see or follow the trajectory. We can obviously also quantify that. And with a quantification, we can then see which transcription factors might have induced the epsilon lineage. In fact, we just correlate gene expression with the density of the pullback distribution. I'm not going to go into detail now because that takes too long. But this way, we predicted NeuroD2 to be an activator of the epsilon lineage. And then we performed immunostaining of NeuroD2, or like of human iPSCs, which had a NeuroD2 knockout, as well as the qPCR analysis, and indeed found that NeuroD2 is an activator of the epsilon lineage.
Yeah, well, so another question regarding, I guess, the slide, how does this transition probability between cell types differ if you compare it with naive Euclidean distance average between clusters of cell types? So if I use embed and then just do average and per cluster and do Euclidean distance, how does it cross compare with the OT version of the distance between clusters?
Yeah, so then the question is basically, if you take the mean of those cell types and compare them, do we use those distances as an input to the optimal transport problem? We don't.
So you were showing cluster cell type to cell type mapping in the previous slide using OT, and there was a question from Eli that how does that compare if you, instead of using OT, just take your cell types, take the average embeddings, and then use the Euclidean distance of the averages. So how much, I guess that on how much heterogeneity, is it unimodal enough that you can use basically the averages of clusters as distance versus using OT?
Yeah. So I think, this entails two answers. Answer number one, if you consider distances, then you would still want to somehow invert those distances, right? Because you want to say you match things which are close and not which are far away. And then one natural way to invert those distances would be, for example, to take e to the minus the distances. And that would give you an approximation. And this is actually exactly how the algorithm works. What the solution does, it basically takes the negative exponential of the cost matrix, but then it has more constraints because you want to make sure that you satisfy those marginal constraints, which means you want to make sure that each cell, or in that case, cell type, is mapped, and each and the source distribution, and each cell type in the target distribution is mapped onto. So there are those constraints of that optimization problem. But I think the intuition is already very correct. So basically, whatever is close by, you want to map onto each other, and whatever is far away, you don't want to map onto each other. And starting from the distances, you first have to invert those distances somehow. And one option is e to the minus the cost, and this is the first step of the algorithm. So that's already like a good guess, good intuition. Second question is, can we just go back to cell types? So here, very clearly, no. We compare in the manuscript using meta cells, and even meta cells are not as good. And then, yeah, cell types themselves, they have many problems just because, yeah, we weren't often not even sure about labeling certain cell types because, yeah, let's say we can use them for interpreting the solution, but we don't want to have a cell type label as input to the algorithm because that then depends, well, the quality depends a lot on the labeling of the data set.
Exactly. And then there's another question from Ziv. And again, I should have warned about like wanting questions in the middle. And so the question is, how does these results compare with other trajectory inference methods like or other tools?
Yeah. So to keep that short, we have a comparison in the paper. So just please look at the paper. And we found that with the other methods, we cannot detect these transitions. The only one which was nearly as good or as correct about the quantification, also please look at the paper, how we measured these, was diffusion silver time. That was also fairly good, but not as good and not as precise as moscot. It was much, much more fuzzy.
Perfect. And I think there's another question which you may answer when I assume you will talk on balance sport, which is on the migration of cells. How do you account for that?
So probably apoptosis and proliferation of certain cells, for example, if we have cell cycles and stuff. So first of all, if you're interested in the cell cycle, whether we can capture the direction of the cell cycle, I would also ask you to look at the manuscript where we describe it. So if we have prior knowledge about whether a cell might rather proliferate or rather die, we can incorporate that. So basically we can increase the weight of a single cell. This was already done by Schibinger et al. In Waddington OT, and we also have everything implemented in the moscot framework. At the same time, if you don't know about it and you just want to rely on the geometry, there's something which is called unbalanced optimal transport. But due to the limitation of time, I'm not going to go too much into detail there yet. So in fact, there's much more to optimal transport than what I just presented, but unfortunately, we don't have as much time.
Thank you.
But there's one thing which which we have to discuss now, going a little bit again about the conceptualization of optimal transport. So what we looked at now, what we introduced was Wasserstein optimal transport or linear optimal transport, which is the most natural, maybe the easiest way of doing optimal transport, because this is exactly when both the source and the target distribution live in the same space. And hence we define our cost function as a function between a sample in the source distribution and a sample in the target distribution. So an example would be we have gene expression at an early time point and gene expression at a later time point. And we want to find the mapping in between those two. And here again, as we discussed, we can take different cost functions. But the nice thing is we can define a cost. Whereas now we look at the middle column, the Gromov-Wasserstein optimal transport formulation. And this is concerned with comparing probability distributions which don't live in the same space. So let's say in Euclidean geometry, let's say you have RP and RQ, R2 and R3, for example. Even in R2 and R3, you could say there's some way to define distances, but for the moment, we just assume we don't, because we could also have completely incomparable spaces like gene expression and attack spaces. So what we have to do here is we have to define two cost functions which measure the pairwise distances. So one's in the source distribution, C1, which just compares xi and xj, both belonging to the source distribution. Whereas, this is a typo that I just saw, C2, which acts on the target distribution, or on the space of the target distribution. So we would compare yi and yj. I'll give you a little bit more intuition in a second. But to wrap these different formulations, we also briefly look at the fused Gromov-Wasserstein optimal transport formulation, which is a mixture of both. So parts of your data points live in a shared space. So this is a little bit like the Wasserstein optimal transport, which is here expressed by C3. So parts of xi and yj we can compare with a cost function, C3, which captures, both the source and the target distribution, but there are also parts which don't live in the same space. So it's literally just a combination of the other two formulations. And an example here would be if we have spatial transcriptomics, and this is what we're going to talk about in a minute again. So that we have two spatial transcriptomic slides, both capture the same gene panel. So this is what we assume for the moment, but we cannot directly compare the spatial coordinates because the two different coordinate systems are not directly comparable because they might have been rotated or they might have been warped. So we cannot define a distance between a coordinate in the first spatial transcriptomic slide and a coordinate or a cell or a spot in the second spatial transcriptomic slide. So I'm not going to go in detail how these works, but again, very briefly, the intuition of how we solve that actually is, again, we have one space which captures or like where the source distribution lives in, and one space where the target distribution lives in, and we want to find a mapping, a coupling, or basically just a match between the source and the target distribution. So we basically want to see whether we could match now S1 to Y3. And therefore we compare the relative positions. So we compare X1 to X2, but then also to X4, X3, and X5, and see whether these distances are similar to all distances in the target space of Y3. So we would compare Y3 to Y1, Y5, Y4, and Y2. And if the relative position, again, measured by the distance to all other data points in the corresponding distribution are similar, we would match them. And this way, we would, again, just fill the transport matrix. Again, here we use entropic regularization, which unfortunately doesn't render the problem necessarily convex. In fact, if it's strongly entropically regularized, it's almost convex, but still this problem is a little bit harder to solve because in general, it's not convex. So which applications do we have? Where do we use that for? So here we have an example of mapping CITE-seq data to space. What we have is CITE-seq cells and CITE-seq readouts, hence dissociated cells. And we have a reference spatial transcriptomic slides. And here we see that we have a partially shared space, namely the gene expression. So the shared gene expression, or like the genes which are also captured in the spatial technology. Whereas we have these incomparable parts of the data, which for the CITE-seq data would be all the genes which are not captured by the gene panel and the spatial transcriptomics technology, and the proteins. On the other hand, we have the spatial coordinates in the spatial transcriptomic slides, and we now match those two cellular distributions by once minimizing the distances in terms of the gene expression, which we can directly compare as it lives in the same space. Obviously, there are batch effects and everything, but still we can compare it, while we want everything which is close in the non-shared gene expression space and close in protein space at the same time be very closely related or be matched to something which is close also in space. So the intuition again is for the quadratic term, or like for the Gromov-Wasserstein part, cells which are close in gene expression space and protein space should also be close in space. And this helps us for two use cases. First of all, we can map markers which are not captured by the gene panel. In that case, we do have this marker ADGRG6, which is a partial lean marker, which is relevant for levizonation. And hence, it helps with the task of levizonation. At the same time, in that case, we didn't have any cell type annotation for the spatial transcriptomic slides. We can also use our mapping to infer cell types. And hence, we can now visualize the spatial transcriptomic slides according to the labeling or the cell types which we inferred from the mapping. And indeed, we can actually see that it makes sense. So this is one example of how moscot, or just generally optimal transport, which was originally proposed by Mornizan, for that use case, can be used for connecting dissociated single cells in spatial transcriptomics. Now, because we don't have much time, I'm going to go to this task which I was alluding to before, which is aligning spatial transcriptomic slides. So this is actually the reason why I said we cannot compare spatial coordinate systems and spatial transcriptomics in general, because those coordinate systems are rotated or are warped. So here we consider the use case of coronal sections of the mouse brain. We have 3 different mice, and in order to get like a more comprehensive view of the actual spatial structure of the mouse brain, we want to align them. And now we do that again with optimal transport. We want to match cells which are similar in gene expression space, and because they live in the same space, we have the same gene panel for both of them, we can compare them directly. So this is like the linear part, the Wasserstein part, whereas the 2 coordinate systems we cannot compare, where we have to fall back to the quadratic, hence the Gromov. And we have 2 options of how to align those sections of the mouse brain. So there's once the affine alignment, which just learns an affine transformation, so rotation and displacement. And here you can see within the rectangle, so we have certain characteristics of those single slides which are maintained. Whereas instead, if we choose to have a warped alignment, we enforce the structure to be similar to the reference structure, in that case to the blue spatial transcriptomic slide. So for example, for the orange one, you can see where there was a sort of a dent on top. We don't have that anymore in the bottom. Where there was no hole on top for the green one, we now had a hole in the bottom, because the reference slide, which is the blue one in that case, also has it. So we want to warp it to exactly match the shape. So yeah, these are examples of how to use moscot for spatial, for aligning spatial transcriptomic slides. Finally, we also present a novel tool for spatial temporal trajectory inference, which is basically a combination of this temporal trajectory inference, which we had in the mouse pancreas example, and the alignment problem. So we have measurements of the developing mouse embryo. And again, here you can see that we capture a full embryo, hence it's very, very large transcriptomic slides, and hence we have to have scalable optimal transport algorithms, because otherwise it's just not feasible to solve them due to their computational complexity. And here we basically, again, apply the same idea. We want to match gene expression, which lives in the same space, similar to the trajectory inference task we had on the mouse pancreas. And we want to ensure that the spatial component now also fits. So cells which are close by in an earlier time point should also be close by in a later time point. So we can again visualize that. So we can look at the original annotation of the heart and the mouse, and we can look at the predicted heart annotation or the mapped heart of moscot, and we can actually see that it fits fairly well. Now, quantitatively, we can also see that the spatial temporal problem of moscot, so spatial temporal just refers to the spatial temporal trajectory inference method implemented in moscot, and temporal, the just classic one without incorporating the spatial component. We see that the spatial temporal trajectory inference method clearly outperforms the temporal, only purely temporal trajectory inference method, which doesn't add the spatial constraint. TOME, in comparison, is a KNN-based trajectory inference method. So yeah, I'm not going to go into details of the differences between optimal transport and KNN, but many people ask still what is different. So there's the differences that we have these marginal constraints, and hence we, in optimal transport, we basically find a mapping based on a global view of the data, whereas in KNN, we always only look at the very closest and hence a local view of the data.
So a quick question from Ziv. What is the technology used in spatial in this case? Is it Visium? And if so, how do you extract cell-specific values?
Yeah, this is CITE-seq. Cell-specific values are bin values. Yeah. So it's just bins, basically.
Okay, It's the bins. Okay. And how do you compute cell transition accuracy? I assume the
That's
one.
That's a very good question. So we basically look at transitions between cell types which are supported by literature. So basically, we have a score because we know that there are certain descendants of certain cell types, and certain cell types were definitely not descendants of a progenitor or like of another cell type. So it's basically like a tree structure. We consider a tree structure of a hierarchy of cell types, and we want to make sure that we remain within the same branch. But that we basically, can directly go from one node to a descending node within the tree. And everything which doesn't satisfy this transition is an error or is a faulty transition, basically. But yeah, please have a look at the manuscript where we define this cell transition accuracy.
And just one more follow-up from Ziv that asked bins of cells. Usually there's a big difference. Wouldn't it be better to work with cell level data if you can come over to the cell level data?
Sure. Yeah. I mean, I think there are different ways to do so. Here, we use bins. Yeah, definitely there are different ways to do so.
And one question from myself, which is, are you restricting your temporal transition from only consecutive time points?
Yeah. So here we basically solve pairwise optimal transport problems. So for consecutive time points, but you can obviously also assess somehow the quality by comparing, for example, concatenated solutions to a direct transport matrix, which we also do in the manuscript. So yeah, please have a look there.
Thank you.
Cool. Nice. So that's actually a good timing because we're going to have 10 minutes for an outlook. So moscot unifies AUT applications in single-site genomics in general. So we have the tools for trajectory inference. We have the temporal problem, which we saw, the spatial temporal problem, which just adds like spatial prior. We also have the lineage problem, which has a prior, which is for lineage, for barcoding data. It's a different manuscript by Marius Lange as well, but we implement it in moscot. We have application for spatial transcriptomics, so the mapping problem for integrating dissociated cells with spatial transcriptomics, and the alignment problem for aligning spatial transcriptomic slides. We also have a translation problem, which builds upon Demetri et al., which basically translates between modalities using Gromov-Hasselstein. And we also offer interface for prototyping, which also take as input and data objects. You have these nice downstream aggregation schemes like the Sankey plots or the aggregated cell type transitions. So if you feel like, yeah, there's a cool problem which you could address with optimal transport, please feel free to use those interfaces. For example, we have a tutorial on how to use the synchron problem, which is a generic problem for linear optimal transport or Wasserstein optimal transport problems, how to use that for perturbation analysis. So just go and check out the website for tutorials and examples. And obviously feel free to contribute. We want to make it a community effort. Then we have a little bit time for an outlook. So let's go back to images. So what if we don't have a perfect match? Just imagine now you have a couple of images with glasses and a couple of images without glasses. So this is actually the CELEB data set. And now you want to somehow match them. For example, you want to have a Harry Potter, which doesn't wear glasses anymore. But we can see that we just cannot find really good matches here. So optimal transport will give us some sort of a match, the discrete solution, because it's convex. We get a solution, we get a probabilistic assignment, but regardless of what it looks like here, we just don't find a perfect match for Harry Potter without glasses. It might seem a little bit odd that we're talking now about images, but I think it's very nice to motivate it because this is just a very good analogy to, for example, perturbation data. Let's say with glasses corresponds to cells in the control state, without glasses to cells without or like with a genetic knockup done. Hence, it's missing a certain gene for example. And this is the analogy, which is why I'm presenting this here. And this is how we can motivate neural optimal transport. So because we cannot find the perfect match in the existing target distribution, we can just say we can generate the perfect match. So here we generated those images with a neural optimal transport map based on flow matching. This is a paper which we had at ICLR last year. But here you can basically see that whenever we don't find a perfect match in the target distribution, we can just try to generate it. And indeed, we generate the Harry Potter without glasses in that case. So neural optimal transport is, relatively small, large, whatever, depending on what you compare it to, field of research. And there are different ways of parameterizing these optimal transport couplings. So we also asked what's actually missing to make moscot neural, which means we don't want to have as output a stochastic matrix, a bistochastic matrix, which tells us which cell in the source distribution map to which cell in the target distribution. But we actually want to have a neural network which generates a cell which somehow looks like a cell in the target distribution, but from a specific cell in the source distribution. So we basically have to learn a parameterized coupling. And hence we looked at what do we need for that. So which properties of optimal transport do we leverage in moscot? So the necessities are, first of all, we want to map stochastically instead of deterministically to account for uncertainties in cell evolution. So as I said, we could also define a deterministic map. For example, the images which I just showed you before, the way we generated them, these were deterministic, but we want to have stochastic mappings because this way we can basically assess the uncertainty of cell evolutions, for example. And indeed, this has been worked on quite a bit already because it also has some nice links to diffusion models. It's something called Schrödinger bridges. Then the second necessity would be the unbalancedness in those entropic or stochastic OT couplings, which we need, as also someone of the listeners alluded to, we need for sequencing biases or proliferation and apoptosis. But there was none unbalanced entropic optimal transport solver yet. Third, we want to have a flexible choice of the cost function to account for this non-Euclidean geometry. We saw that in the pancreas example. This is really relevant. But the existing stochastic neural optimal transport solvers or Schrödinger bridge solvers rely on Euclidean costs or Euclidean geometries. And finally, and very importantly, we want to map across incomparable spaces. So also we want to incorporate this chromophacocystein idea and fused chromophacocystein idea, because as we saw in moscot, most of the problems are actually chromophacocystein or fused chromophacocystein. But this didn't exist either. So in a paper at NeurIPS last year, we introduced GenA, which is entropic chromophacocystein flow matching, and had a few applications to single-cell genomics where we basically just show that what we do with moscot, like trajectory inference, like perturbation modeling, like translation of modalities, we can also do using neural optimal transport. Cool. So yeah, please check the paper out if you're interested. And my acknowledgements briefly to all my amazing co-authors in moscot for the unbalanced MOSMAP paper and GenArt. So I think we have a couple of minutes for questions.
Yeah, thanks so much, Dominik. So I have one question. So moscot and OT problems, I guess, so moscot's version of OT problems is primarily focused on interpretation of observational data, right?
Mm-hmm. Yeah.
Most of the extensions you discuss are extending this to, use maybe observational data for interventional experiments in silico, which is very cool.
Mm-hmm.
But I wanted to go back to the interpretability problem, because what you're doing with distributional matching is sort of saying, take one sample from one distribution, match it to another optimally. But there are structural models like regulatory networks underlying the distribution at both points that I think, for the most part, I guess the classic way of interpreting transcriptomics would be to just infer a regulatory network and say this is the model, the parametric model interpreting everything. But this is a problem with optimal transport because we no longer have like a parametric model that's defining the distribution at either point, right? Source or target. So I'm wondering if you could talk a little bit about like, how do we interpret what is happening from source to target with moscot, right?
Yeah. So yeah, interpretability indeed is a limitation. So I guess in general, no model is perfect. And also it's very nice to have these parametric models, for example, RNA velocity. But at the same time, we know very many failure cases of RNA velocity. And hence, it's just a different approach. I wouldn't say one is better than the other. So it probably depends on the tissue. And there are also failure cases of moscot and especially limitations of moscot, as I just mentioned before. If you just blindly apply the Euclidean cost, it might not work. But how we try to get interpretability is, so yeah, depends on the application, of course, but mainly we look at the correlation of features like gene expression with a certain pullback or push forward distribution. So basically whenever, let's say, the way we found NeuroD2 was we literally just, correlated the density of the pullback distribution. So basically we looked at the row of the optimal transport matrix and correlated that with gene expression and looked at, which gene expression or like which gene is expressed in a very similar way as the density of this precursor population, which we hypothesized. And this way we look at what is the driving factor. Similarly, we can identify target genes in a very similar way of certain transcription factors. We basically correlate the distribution which we predict from a certain population which highly expresses a certain transcription factor, which correlates a lot. So we tried to include a couple of ways, but indeed there's no parametric formulation of this which incorporates like gene regulatory networks or so.
Yeah. Well, I think co-expression networks are a perfectly fine parametric interpretation of the gene regulatory patterns. I think that's very interesting. So you actually don't need to infer the correlation matrix on source and target. You just need the source and the push forward, is what you're saying. Yeah. That's very nice.
So some people, like if you have the gene co-expression matrices, they typically rely on a Riemann manifold. So you can define metric distances on Riemann manifold as your cost for optimal transport. So instead of point to point in a VAE space, each point may be presented by the full co-expression, and the cost matrix is the cost on a Riemann manifold before you do alignment. And that also people have been exploring some of that helps you with interpretability because you're mapping basically matrices to matrices of co-expression. Thanks a lot. We are at time. I just wanted to thank you a lot again for a wonderful talk. There was probably one more question. Let me see, and that might be the last one we get. And then you could infer push forward from local environment or global. I don't know if that's a question or answer. Okay, so thanks again, and we really appreciate it. The edited version is going to be also up online for people who didn't make it. And at this point, I think we are going to conclude this talk. Thanks again.